Hey y'all. Happy Tuesday. No, dang it. It's Wednesday. You know what? Y'all, life is hard. I cannot keep the days of the week together. No one I know can. Like, I really only know whether or not I need to teach like one class or my other class. So luckily we haven't shown up here and I've tried to like draw some, Gen Chem stuff on the board. But today is Wednesday. Self-care is, as usual, difficult. I did go for a run today. Been trying to go at off-peak times, for social distancing. I ran today at a little bit before noon. It was very humid outside. Now, I know that it's Florida. That's not new. That is not. It was a struggle run. Got out there, just trudged along, and that's okay. I got out there and I did it, and it was good enough. Lily is not here today, unfortunately. Frankly, she's over going to school, I think. I think we've disturbed her naps for enough weeks in a row, and so she's napping. She might come in later. It's kind of hard to say. I thought she was going to come to class and then was like, no thanks. But because I felt like my announcement slides on Canvas were getting a little, hey girl, a little boring, I have provided you with 2 new animals today. Phoebe is a cat. She is named after Phoebe and Friends, and her dog sister, Harper Grace, who's a red golden. These are my friends' dogs, and they're just so fun. Thought you guys could have more things. But they are cuddling together on a rug, sleeping. And Lily has been a little like, no more photos, please, please. None of that. So, couple of things. So I think on Monday, I want to say it was Johnny that asked, whether or not med schools or professional schools were going to not accept, but allow you to earn a satisfactory or unsatisfactory in a course on your transcript and still be accepted. So yesterday I chatted with Dr. Nuckley and asked like, hey, what you got here? So he and I both have the same idea, for lack of a better term, that because what we would call aspirant schools, so the University of Tennessee, Texas A&M are also offering these, you should not be disqualified from admission by earning a satisfactory. So it was a common misconception with my Gen Chem students that whether or not if you opt in, you would get quality points. So on I copied the wordage. So on the undergraduate studies website, in this system, so if you opt in, students will be eligible to keep all A's and B's, but C's will be converted to satisfactory and D's and F will be reported as unsatisfactory. So Biochem 2 is a terminal course, meaning we don't have Biochem 3. Don't I wish we did too. So the lack of Biochem 3 in that case will allow, if you get in a satisfactory here, it would count towards the next course. So if you're taking, I don't know, Physics 1, you can earn a satisfactory and still go to Physics 2. I don't actually think any of you are taking Physics right now or Physics 1, but hypothetically, if that was you, that would be the case. So other things, your figure analysis should be graded by the end of the week. I will grade those in Canvas. It will show up as a populated score in there. So homework 3 opens at the end of class today on Canvas. So the way this will work, it's 2, I think it's in its own module. So what it will do is one of them is a downloadable link for the homework packet, essentially, which would have been what I provided you in class. So it should look just the same. Doesn't count. So the only thing that doesn't count is an S or a U. So basically, the end all be all. As I, Hannah Renee Malcolm, understand it, so you cannot use this in like GPA court, if that's a thing, is that the A and the B will count for you, but it is a way for there not to be a C, and the C will not help you or hurt you in your transcript. So if you opt in for any course and earn an A or a B, that will show up on your transcript. If you opt in and earn a C, D, or F, you will not see those letter grades. What you will see instead is for a C or C+, you'll see satisfactory. For a D or F, it will say unsatisfactory. Neither of those or against. Does that make sense? So homework 3 opens today. It is a PDF document. Now, for those of you who have a tablet and you can upload the PDF, feel free to write on it. You can, and I like to use, not Notability. Notability and the other one will both allow you to do that. I think you can write on them in Canvas. So you can edit the PDF, you can use text boxes on there, whatever you'd like to do, and then upload that back up as a PDF. Now, I'll be honest, I own an iPad, but I do not own a printer. It turns out that I have never needed a printer in my house. I just print stuff at work. So if you are like me and you do not own a printer, hopefully you have paper at home. You can do it on the back of something else. I don't really care what you do it, but please annotate your homework, like clearly mark number 1. You don't need to copy the question. You can just answer the question and then draw a line underneath or some other way for me to know number 1 is different than number 2. But please do not feel obligated under any circumstance to download and re-upload, like to buy a printer, go to UPS to print something. You don't need to do that. You can do that and then cam scan your pictures and upload them. That's kind of up to you. Your homework will be due next Wednesday at 4:30, all through Canvas. If you have problems, I'm happy to help you. Homework is the same rules as before. You're more than welcome to work in groups. I don't want to say we can all work in a group together, but if you have questions, I'm happy to answer those. That opens today at 5:45. And now that I think about it, there is a small possibility that I forgot to unlock that module. So I will look at that as soon as class is over to make sure that that's available to you. But that is what's coming. And so the molecular techniques, we're going to keep doing that next week. We may add some more. Just going to kind of see how it goes today. So any other questions before we start to talk about what do we do next now that we know that we were able to fully sequence our constructs, so we know that we've created a protein in our vector. I also mentioned that we were starting with RT-PCR and an Amari Povich kind of way, like that was a lie. And so we're going to actually start with SDSPAGE and Westerns and some protein expression techniques first, before we talk about RT-PCR. At the end of class on Monday, we have a sequenced gene plus vector. And so if you remember, our vector map looks something like this with lots of different colors on it. We have our gene hanging out in here, and we still have our AMP or other resistance cassette. So what we want to talk about today is how do we make protein? So the first thing we're going to talk about is the induction of protein using the promoter. So if we think about a vocabulary word of induce, so to induce something, to selectively turn this protein expression on. And so we want to do that using this promoter. So we always drew this red thing. So the red thing is our promoter, and this is part of the vector. So when you go to the science store, you can determine whether or not the vector you have is different promoters promote in different ways. So we'll talk about two different types today. So we're going to talk about the arabinose promoter, so all of them are going to be quite similar to chapter 26. So if you remember chapter 26, which to myself feels like I taught it to you like 18 years ago, chapter 26 was all about gene regulation. So you might remember the lac operon. So one method of promotion, or most of them fall into this category of small molecule inducers. So one of those is IPTG. So isopropyl thioglycosides, I think. Yeah, that's right. Isopropyl thioglycosides, or glycoside. It is a small sugar molecule that works like allolactose make sure you guys can still see. And so this will turn on protein production. So typically, one of the things that we want to think about in a biochemical assay is how do we selectively turn on protein expression? So we don't necessarily always want our protein to be expressed. Sometimes you do, and that is a different set of experiments. But in this case, we want to express the protein either selectively or only at a certain period of time. So first we're going to talk about first we're going to mostly think about an E. coli. So there are ways that you could, if you added your plasmid to a mouse model, there are ways that you can selectively turn on genes within like a furry little mouse or a non-furry mouse, no judgment here. But you can add the mouse to any of those situations or the gene to the mouse and express them in that system. But mostly for the purpose of Biochem 2, we're going to talk about the induction using the promoter inside the cell. So typically, we want to grow our cells. So typically, we grow our cells to we're going to call it a medium density. Basically, this means that for E. coli, we have a growth curve. So on our x-axis, we have time. On the y-axis, we're going to have the optical density. Optical density is a visible spectroscopy method where we can basically measure or extrapolate how many cells are within the volume that we're looking at. So E. coli tend to double every 20 to 35 minutes, depending on the strain. So over time, we should see something that looks like this. Where down here, this is often called the lag phase. This is the logarithmic, the log phase. And this out here is the steady state. So a couple of things about these different regions. So when we think about the lag phase, the reason it's called the lag phase is it takes a while for things to get moving. Now, my least favorite math questions when I was, last week, were the questions of, if your money doubles every day, on what day do you have $500,000? So it's the day before because it doubles every day. So we collide double the same. So down here in our lag phase, what's happening is one cell becomes two cells become 4. And the measurement, the ability to see this is quite low. In the log phase, the optical density rapidly increases. We want this region here. This is the ideal time to induce. Now, I will admit, as I often do, I am teaching you guys my personal weird lab voodoo. Different people like to induce in different places. I prefer to be at the early of the log phase. I have also done things where I induce much later. It kind of depends on what your ultimate goal is. But the point is, in this moment, we're going to add, So we're going to add IPTG at this point. And so as the cells continue to divide, they will begin to add energy into protein production. One thing that you might remember from class is that to make a protein costs energy. So if we want to churn out all of this protein, it's going to require ATP, molecules of interest, amino acids. So we want to add IPTG here. At the steady state, the cells will continue to express protein. It will make similar levels of protein. So from here, we've induced our protein samples. So now we have a cell, and inside we have the plasmid and the protein. So I work on what's called an insoluble protein, meaning it's in the cell membrane. Most people work on soluble proteins that are freely kind of wiggling around inside our E. coli. That's fantastic. We're going to mostly talk about those type of things. I will admit that protein purification and expression is often wrought with difficulties, meaning this can go wrong. You could have a protein that could kill your cell. For the purposes of right today, we're going to talk about this when it only goes right. Because it would take us another year to discuss all of the different ways that this could go wrong all across the board. So now that we've expressed our protein, now we want to be able to purify our protein. So likely last semester with Dr. Knuckley, you talked about SDSPAGE, and you might have talked about Westerns, but we will talk about them again because I think they're important. So the next thing we want to start to think about is protein purification. So the thing is, inside our cell, we have, today our protein, like our gene, will be blue. So we're going to have lots of blue particles. Oops. But also, IPTG mostly only works for parts of the lac operon. I think there are some novel things where IPTG will also induce those things that have been lab generated. So I was asked, what would be consequences if you added during the steady state? So if you add during the steady state, cells are no longer really dividing. They've kind of reached what I don't want to call it affectionately, but in that case, they're not doing a lot. And so one issue that you run into is if they're not rapidly multiplying, you do run the risk of the cell being in a state where it does not produce any additional protein. So in steady state, what leads to steady state is also a nutrient depletion. So steady state arrives when the colony has basically become as large as it can be in the space that it can be. So if you induce at steady state, one of the issues is we'll only get protein out of those cells. And so you get biomass. So in that case, now there are people that like that phase. My trainers years ago prefer very early in the lag phase. And so one risk of doing that is that you will not get as much protein as you would in a lower phase. The other issue is that they've started to consume all of the nutrients. And one of the things we learned in chapter 26 is once you get up into that steady state, you start to turn on other biological processes like the CRP-CYA process. So CRP is the cyclic AMP response protein. And so in that case, it's going to start to tell the cell to make other metabolites. So it now has to spend most of its energy surviving instead of its energy producing protein. And so if it can choose, it will choose itself, which makes sense, right? So the cells will only put in what leftover energy they have after cellular survival. Does that make sense and answer your question? I can answer that. So if in our cells, we have our blue protein, we're also going to add in some other purpley proteins. And now I'm going to add some dark green proteins. So the question is, well, you can't see any of that, so you'll just have to trust me that they're different colors. But one of the points of protein purification is, or our goal here, is to selectively purify the protein of interest. So we need a way in order to select for the blue proteins. Now, my personal preference with that is to use antibodies. So we'll call it the HRM approach. Is to use antibodies or tags to purify proteins. Now, the tag that we're going to use in this case is called a His tag. And so if we were to draw our blue protein here, here's our protein of interest. And hopefully we can see this color. If not, I will try again. I mean, you can see that there's something on there. So this is what's called a His tag. Now, His tags are that. Nope, no one can see that. Hold that thought. So a His tag means that you have 6, well, we'll say 5 to 9 His residues together. So I like to put mine on the N terminus. You could put yours on the C terminus. It is very rare for your histidines to be somewhere else. And you can put them, they need to be all together at the end or the beginning. And those will often also be encoded in the vector. So this His tag is how we're going to purify proteins. You can also use, there's something called an S tag or an HA tag, or you could use GFP. GFP is a little bit harder, but here, this tag is going to give us a way to select for this. So what I want you to picture in your mind, and I wish I had a demo. If I gave you a bowl of, we're going to call them Cheerio products. So we have regular Cheerios. We have the new cinnamon Cheerios, pretty good, and Honey Nut Cheerios. From afar, like from here to you, they're kind of all going to look like Cheerios, right? But I only want the plain Cheerios. And I don't tell you how many there are, which means that you have to tell, okay, you can do it visually, right? Plain Cheerios are not shiny. So you could do it that way. But if you could imagine a way for you to, let's say, this would be gross, but if the plain Cheerios were magnetic and you could just stick a magnet in there and they would all suck to the magnet and you could pull it out, shake them off, and do that again, you could easily and selectively purify the proteins of interest. So this His tag is going to function kind of like a magnetic tag. So we're going to do something called affinity chromatography. So affinity chromatography, which no one can read, so let me rewrite that. This is going to allow for the selective purification of a protein. And so this affinity chromatography is going to allow us to selectively purify the protein that we care about. So in this case, we're going to talk about His tag proteins. There are slight flavor differences depending on what your affinity tag is, but the overall mechanics of that are all going to be the same. You could think about it kind of like if you wanted to take a proton NMR versus a carbon NMR. You just need to do the setup slightly different, but the way you actually load the NMR sample and collect the data is the same. So that's what we're going to kind of think about today. So how do I want to think about this? I'm going to give you guys a second to ride along before I erase that. So then we'll walk through the steps with this, and then we'll talk about putting it on an SDS page and using a Western to verify that we actually got the protein that we cared about. So once we've grown our protein, we now want to break open our cells. So step one, lyse cells. And there are about 100 different ways you can do that. It's kind of outside the scope of what we're going to talk about today. But once we lyse our cells, we now have in a beaker all proteins. And so what we want to do is find a way how to selectively purify our His tag. So we want to use for His tag purification, we want to use a column with nickel 2+ on the beads. So now you might be like, nickel 2+? Why? So histidine residues are aromatic pentameric ring systems. So they look a little bit like ferrocene. So if you remember back to inorganic, ferrocene is 2 pentameric. I think it's pentameric. But basically we want these nickel to correlate. No. Yeah, we'll go with correlate. Correlate to these histidines. So you want a lot of histidines in a row. So then you're going to be able to take all proteins. So one, you load all protein to column. So then what we should have is the proteins of interest should be in here. And we're going to add red proteins. We don't have any red proteins, so we won't do that. Never mind. We're going to use blue proteins. And this is purple. So we'll add some green. So all are on here. So our second step is then we want to wash any non-specific proteins. So we can do that, it turns out, by just flowing buffer through. So in here, we're going to have our wash. And in there, we're going to get some green. And most of the purple, or some purple. So now the trick is, once we've stuck the blue stuff to the column, how are we going to get it off? It turns out that we can start to elute with, in this case, imidazole, or any other competitive binder. So the idea here is in the wash, we're going to remove non-specific. So if you think about the magnet and the Cheerios example we talked about a little bit earlier ago. Yeah, we'll talk about that in a minute, Ryan. So if in this case, we can remove anything that's non-specific, stuff that just kind of hung out, but it's not really well stuck to the column. So then we want to start to elute off. This is our, can you see that? Yeah. In our elution fraction, we'll just say our eluted, what we expect to find is only the blue protein. So the way the his tags and the nickel interact, so if you have your backbone with your histidine rings, which are aromatic pentamers, right? So if you have a nickel 2+, you should expect that your histidine rings can chelate, so the his residues chelate to nickel 2+. So the nickel 2+ chelates to the histidine residues. So the reason imidazole, which is basically just the histidine like side chain, chelates to that. And so in that case, the elution is a competitive elution where that molecule competes off the other components. So I know that Dr. Nuckley talked about this last semester. It was in the protein techniques lecture, which is right after chapter 6. I will post the pages out of the book if you have more questions about that. So in this case, we were able to purify our protein. And so we talk about this because if we want to continue to ask questions about it, for example, what does this protein do? I only want to know who it is, right? So I want to know, do I have only the protein of interest? Do I only have, we named our protein Apple Jacks, do I only have Apple Jacks in the bowl? If I have Apple Jacks, Corn Flakes, and Frosted Flakes, and we get something special, how do I know what protein is responsible for that? So in this case, this is affinity chromatography, and affinity chromatography basically takes all of our proteins puts them on a column, and then uses a competitive binder. So basically, if you think about musical chairs is a little bit how this works. So all of your proteins of interest stick to the column, which is full of nickel 2+. Eventually, you wash away anything non-specific. From there, we want to elute with imidazole. So really well, imidazole chelates super well. And so when you do that, the imidazole then outcompetes the his column, the his tagged proteins to elute out. Any other questions before we start to talk about Westerns and SDSPAGE? So SDSPAGE is sodium decasulfate polyacrylamide gel electrophoresis, otherwise known as just a protein gel. So this is going to allow us, so we can sort protein by size. So what we typically see in a SDSPAGE is, we would call it a protein gel. Down here, we will have small proteins. At the top, we will have large proteins. And so what you should see is, based on your wells, you can have different types of proteins at different quantities. So maybe in this lane, you only have this protein. In this lane, you have this protein. Or maybe you have something that looks like this, which, to be quite honest, is a nightmare. This means that you have so much protein that you can't actually select. So the trick is with SDSPAGE, proteins are clear most of the time, unless you have GFP on them, and then sometimes you can see it on a gel. But by and large, our proteins are clear. When you have so much protein, it can start to achieve a color. So maybe you have a protein with a fluorophore in there or something else. You could start to see it that way. But in this case, we need to be able to visualize this. So we can visualize the protein through a non-specific dye molecules. And so in this case, any protein that is found in the protein will turn, the one I like to use is blue. There are also a couple of new ones that will turn it red or green. Blue is the most traditional. So an SDSPAGE is going to allow you to sort by size. Now, it does that. So, proteins are sorted by charge. So SDS molecules, so sodium decyl sulfate, is basically a sulfate head followed by a long chain. So the SDS molecules interact with proteins in so many charged molecules per size. So each protein binds a SDS, binds SDS in a ratio that, make sure you guys can still see this, that allows size to charge sorting. So typically when we think about separate by size, we think about migration through the acrylamide. So agarose is based on size and charge. So the smaller molecules can go faster. That is also true here. However, we're going to use an electric charge to push our proteins from the top of the gel to the bottom of the gel. And we do that based both on their size, how fast they move, electric charge to something that is. So mine just said it buffered. So I'll back up a little bit. So proteins are sorted based on size. But if you apply charge to an uncharged molecule, it turns out that it won't go anywhere. So in that case, you need a method in order to push the protein through the gel. So we're going to use electric charge. The SDS in this is found both in the gel and in the loading dye. That will then interact with the protein. It will then allow it to migrate down based on a size to charge ratio. So if you do not do SDSPAGE, you can do this. It's called isoelectric point focusing. We are not going to talk about it today, but you don't have to use SDS. It works a lot better if you use SDS. So when you don't use SDS, the size to charge selections are a little bit difficult to determine occasionally, because you have to know how many charged residues you have corresponding to the size. So it's slightly different. So from here, what we've been able to see in our gel, so if this is our ladder, some way that we can say what size it is. This is our ladder. You can't really tell that, so we're gonna just make that bigger. So we have a ladder. This is basically a ruler that we can then say, so the ladder sorts by size. So Johnny asks, if you don't use SDS, you can't sort the proteins with electrophoresis at all. So you can sort with electrophoresis. You sort by the isoelectric point, so you just can't sort by size. So a better example would be if in our bowl of Cheerios, so we'll go back to our breakfast cereal. So if instead of asking, so by size means that we have Apple Jacks, Toasty Oats, Cheerios. We could basically sort those using a funnel by size. So in this case, the question becomes, what do we do with, is basically the isoelectric point is the point in which the protein is neutral. So you have to know the isoelectric point in order to be able to sort based without SDS. So you can. It just, I'll try to send some, I'll try to make something else. I'll try to send you some documents about what some of these look like today. Basically just a PDF of here's this, here's that. Because I know that my drawings are real amazing, but they're not that amazing. So the question is, with that in an isoelectric focusing or a native page, it's actually really hard to figure out what it is. So you can sort. You can't really sort without SDS, or you can only sort by other factors. I feel like that was a very long statement about how I didn't really answer your question. So the short answer is you cannot sort by size without SDS. You can sort by other methods that you've investigated in that process, for lack of a better term. So the next thing, so we have a ladder that gives you the ability to determine what we have. So, but the other question is, if we have two different bands, we have this upper band, so a heavier protein or a lower protein, how am I going to know which is the protein I actually care about? So it turns out that SDSPAGE just tells you, hey, you have protein. Now, likely, kind of like if you collect an NMR, you have a structure and you know what you think it should be. The trick with SDSPAGE is that it's super non-specific. And so you can just say, like, 50 kilodaltons. Okay, well, this band is roughly 50 kilodaltons. Good enough. So sometimes we would like to use another more in-depth technique in order to say, this is my protein. And so the protein that we're going to use to do that is what's called a Western blot. So Western blots are a way where we can say with most determination that, hey, this is the protein that we want. Oops. So Western blots analyze SDSPAGE for, and this is the key here, specific proteins. So the first thing that we're going to do is we're actually going to, we need to transfer the proteins from the SDS into the Western blot. So the way you would do this, if this is our blot, you're going to lay your SDSPAGE on top of it. So we're going to take the SDSPAGE you just ran. Before you dye it a color, you're going to lay it gently on top of a blot paper. So this is either a nitrocellulose membrane or a PVDF membrane, basically a membrane with very small holes, because you want your protein to go onto the blot, but not through. From here, we're going to apply a voltage. So in order to isolate or to separate by size in an SDSPAGE, we applied a voltage. Now we're going to basically set it like a pancake. So we made, maybe not a pancake, you've made a sandwich where we have one electrode, the blot, the SDS, and another electrode. There are, in fact, some buffering membranes here. I've left them out for the purposes of drawing clarity. So from here, when we apply a voltage, the protein will go into the blot. So your protein migrates from SDS to blot. So once you've gotten the protein out of the SDSPAGE into the blot, we're going to be able to analyze that. So we're going to apply a voltage. It runs to red, and so black leads are I think they run to the negative side. Don't quote me on that. That will not be on the exam. But in this case, we're going to push our protein out of the SDS onto the blot. Now, we're going to be able to account for all of our proteins to basically be pushed at the same rate. So in this case, we're not going to sort by size. So all of the proteins are exposed to the same voltage on top of them, and it basically pushes down into the blot. So we basically transferred the proteins out of the SDS into the page, or out of the SDSPAGE into the blot. So we've done that so that it is on a surface. So SDSPAGE can kind of be flimsy. This is kind of like, I don't want to call it wet paper because that sounds terrible, but more of a firm wet paper. So that doesn't really make it any better, but we're going to put all of our proteins onto the blot. So once we've done that, we want to be able to ask, is our protein in the SDSPAGE? So now, once we've transferred the proteins to the blot, we're going to start. We're going to analyze the proteins, which I really should have not written down here because I'm going to have to write it at the top. So we can talk about analyze these proteins. So it turns out that our proteins are bound into this paper. So here we're going to have two proteins. So this is going to be protein 1, and this is going to be protein 2. These are just any proteins. So one of them is going to be the protein we want to see, and the other one is going to be a protein that we don't care about. So a Western is going to only answer one question. Is my protein there? Yes or no. It doesn't tell you about purity. It doesn't tell you about environment. It just tells you, is it there? Yes or no. So Westerns are a really great technique for, I have a lot of proteins and I want to know if my protein is there. If you want to know if your protein is pure, you're going to need to use an SDSPAGE in addition to the Western. So the first thing we want to do, so the analysis here is going to use an antibody. So in Biochem 1, I know I talk about antibodies. They're really popular right now. So one of the new, I don't think this test has been marketed yet, but one of the things they'd like to do with the COVID-19 is to use an antibody test. That antibody test basically takes a serum sample, otherwise known as blood, for those of us who don't like to think about it. So if they're going to take a blood sample and then they're going to go in there and ask, have you had an immune response? So we are going to use antibodies that have been generated to detect different things, not COVID-19. So we're going to use antibodies to detect the protein of interest. So you can develop an antibody. So option A is a specific to protein. Option B would be specific to, in this case, we're going to call the tag. So if you remember that we purified our protein via his tag, we can also use an antibody that has been raised to a his tag. Now, I'm not going to draw this because it gets kind of weird. So the way these antibodies are made is we inject an animal. So you can do it with chicken eggs. You can do it with mice, rabbits, goats. I think I've seen a horse. So basically you can inject this animal or animal cells with this antibody, with a protein. Once they are injected, they're going to get an immune response. So if you think about vaccines, which we're not going to talk a lot about, but vaccines use dead virus in order to tell your body to create an immune response. So we can inject in this case, we're going to take an animal, a rabbit, we're going to inject it with a specific protein. That specific protein is going to raise an immune response. So now in their body, they have antibodies that directly correspond to the protein of interest. So now if we get these two things in the same space, they should interact with one another. So what the antibodies do is they basically create a binding interaction, a little bit like Velcro. And now they're stuck together because they're perfect matches for one another. So in this case, these two, either of these are what we call our primary antibody. And it detects the protein. Whether it is specific to the tag, which allows you to buy one and look for multiple proteins in different environments, or specific to the protein. So in this case, our primary antibody is going to be red. So in this case, we care about protein 2. When we add the red primary antibody, it's going to interact here. So we now have our primary antibody lane. Primary antibodies are often indicated as a number 1 followed by the degree symbol. It's just the way it's always been, I guess. So our primary antibody directly detects P2, but you'll notice that it doesn't bind at P1 at all. That's great news. So now we could selectively say, hey, we see P2. But it turns out that it's really just a one-to-one interaction. Protein hanging out with the protein antibody. So what we need to do next is we need to amplify this signal. So we're going to then look for, So we want to amplify the primary antibody. So this is what we're going to call our secondary antibody. And it detects the primary. So our secondary antibody does not under any circumstance detect the protein. So our secondary antibody, which we'll use blue because those colors seem to work, you can get lots of binding to each site. So you'll notice that we had 2 primary molecules, and our secondary looks like this. So now we went from a single protein to 2 primaries, which each hold 3 secondaries. And now basically we're amplifying the signal. So what we've done is we've taken our 1 protein and interacted with more. So the secondary antibody is made the same way as the primary, except oftentimes they detect the first host. So this would be anti-His, maybe we got it of the chickens. The second one would be anti-chicken goat, meaning that we use the goat to detect chicken antibodies. But the other thing with the secondary antibody is it has another part. So it has this, but it also has I'm gonna have to erase up at the top and that way we have enough space. So the secondary antibody also has a detectable part. So the one I'm most familiar with is horseradish peroxidase. And when you add a small molecule, upon activation, it gives off light. So when we activate it, we are now going to get all of this green fur here. So basically, when we added, so the primary antibody detects the protein. The secondary antibody detects the primary antibody. The secondary antibody contains a small portion of it. So in this case, it has a small HRP sensitive molecule that when you add horseradish peroxidase, cleaves the bond and it gives off light. This is how we can detect in a Western blot. So typically Western blots are used for a couple of different things. Do I have my protein? Yes or no? So we will start to talk about after this. So if you have questions, go ahead and write them in the box. We'll talk about other ways that we can look at a variety of conditions to ask questions about how do different proteins interact. So we could use this to say things like, how do proteins interact? So right now we've just talked about what happens if your protein is purified. You can run a Western and say, you have your protein of interest. So the next two topics are kind of tied together. We're having reconnecting issues in case this is actually working. So I think it kind of buffered. It probably did. It buffered on my end. So what we want to think about now are ways that we can detect changes in both expression level and what happens in that case. So Ryan asks, what if the desired protein is unknown so that we don't know an antibody for it? Then you cannot use a Western. So if the protein, so the question is, if your protein is unknown, so far we've talked about proteins that you've purified, which means that you do know what you're trying to purify. So if you're not quite sure what the protein looks like, you could add your primary antibody could detect the his tag. And so that will be separate than the protein of interest. So you could do that. But if you are asking about which protein it is, so if the protein is unknown, you do not, what is another way to put this? If you are the first person to discover a protein, then you cannot have an antibody that binds to that protein. If your protein has a his tag on it and you're not quite sure what it does, you can detect the his tag with the primary antibody. So the primary antibody can either be tag specific or protein specific. So Johnny asks, how do you know what antibody to use for a protein? So the shortest answer is there are a couple of different ways that you can get antibodies. Option number one is you can detect the tag. Option number two is if you can purify enough of your protein without a tag on it, so just your protein of interest, you can actually mail it to a company and they will make antibodies for you. Y'all, that's real spendy, but it works really well. So the other thing with this is we often talk about going to VWR or Fisher. It turns out that there are different biochemical companies that basically make books. And book is really strong. A light, significant magazine. And I want to say it's Jackson Labs is probably one of them. Jackson Labs is one of the places you can buy this. But you can go in there and basically look at it and say, hey, all of these might work. So a lot of times antibodies are raised against specific parts of proteins. So if your protein has a cyclic AMP binding domain, there may be an antibody that's already been raised against that. So you can often go and try things. But a lot of times, in order for this to really work, you need to be able to set up, something about the protein that you're trying to purify in this case. Good questions. Any other questions? So one of the ways that we can use Western, so use number one, uses one, determine if you have purified protein. So option number two is a little bit different. So one of the other things that we could start to do is to compare protein expression under different conditions. So So if we have cells grown into different environments, could we see the effect of how this protein works? Let's think about if we've added some type of an enzyme that is upregulated, or you're upregulating it yourself. So we could start to ask environmental questions using a Western. So we could look at cells grown with all nutrients, cells grown with only some nutrients, only few nutrients, and only essential. So, real life examples of this would be asking questions. This is someone who has a normal diet. This is maybe someone who's excluding maybe gluten-free. You're excluding, you have most everything you need. Also, I'm just picking diets here. I'm not saying anything about gluten-free life. So only some nutrients, only a few nutrients, and only essential nutrients, like that Soylent stuff. So basically, if we compared the athletic ability of someone under 4 different diets, we could do that, and then you could compare, right? So if you're asking me, how fast can I run 100 meters? You know, like in the Olympics. So it depends on how well I've eaten. So if I eat really great, then I'm going to have one baseline time. If I haven't eaten, or I've only been eating lean cuisine or some other light, only the essential nutrients, I personally will have a hard time functioning. So we can grow cells under 4 different nutrient conditions and then compare those on a gel. So one thing you might be asking is, okay, but how do you know that they're the same? So in this case, we're going to have to start using something called a control. Now, controls in biochemical experiments are really important. We've talked about this with Fret before. So with Fret, you can now say, like, okay, how do these interact? How do the other ones interact? But you need to know what should you see or what should you not see. So in this case, we want to also use a protein control. So a protein control in this case is going to be one of our more favorite proteins, housekeeping proteins. So housekeeping proteins, or proteins that are expressed at the same level all of the time. So this would be something like a constitutive protein. And so what you can start to do, what we can see is, what we should see, we want the ability to compare these 4 situations. And the only way that's going to work really well is if we have the opportunity to know that we didn't add more of the protein of interest by accident here compared to here. Because that doesn't tell us anything about our proteins. So up here, we are going to have, I'm going to title these 1, 2, 3, 4. So in our Western, we're going to look for the difference between 1, 2, 3, and 4. So in this case, we are looking at, these are E. coli. We're looking for the CRP, so the cyclic AMP response protein. We expect that to be upregulated when you look at essential. Like if you don't have enough nutrients, the cells will adapt to consume other nutrients. So in this case, we see small amount here, a little bit more here. So in this case, it looks like value 3 has the most amount of protein. I think that buffered. So when you look at this Western, we have, so this is our CRP protein. This is our protein of interest. So what we can see under all nutrients, we have very little. As we decrease the nutrients, we have the same amount. But when you get to condition 4, which is where we would have expected to see the most, we don't have any. So we're also going to add our control. So we're looking at CRP, which is in bacteria. So I'm going to use an unknown control. In mammalian cells, we often use actin or other cellular proteins that are super consistent. So what we can see here is in lane 1, lane 2, and lane 3, for give or take, all of these 3 are the same. So this loading control tells us that we can definitively say this gets bigger as it goes across. So the point of that is that we can increase, we can see that. In this case, for number 4, this does not look the same. So we know that we made a mistake, not as a scientist, like a bad mistake, but somehow there were less cells in here. So we need to continue to investigate what happens here. So Westerns can be used to start to ask questions or answer questions about how proteins function under differing environments. So we can look at this and say that, okay, under our normal nutrients, we see very little of this protein. As you decrease the nutrient amounts into lane 3, we can see that that protein is now upregulated. Upregulated means there's more of it. In this case, we cannot say anything about number 3. So is the control supposed to be increasing from 1 to 3? Shoot, those are supposed to all be the same. Let me write that up here in case you're watching at a later date or without sound. Those should all be the same. It's very difficult to draw like that. I'm a terrible artist. So in these 3, your loading control should be the same or very slight differences. But in lane 4, we can see that the control is not the same. And so we cannot include it. Now, if we were to rerun this entire experiment, nope, let's make that a little bit further out. At 11, why? So this is for B2, and in that case, we see that that is now the same as well. And so in this case, we see an even larger quantity of protein, which tells us that under little to no nutrients, we have the highest expression of that protein. So the control is a measurement of the quantity of cells run per lane. To some extent, that is true. So the point of the control is that you need them to be somewhat similar. So ironically enough, that number 4 control even looks more worse than it should have before. So let me erase part of it. That didn't help. So that one should also be the same. It is a quantity of that type of thing. It's also often used as the ability to ascertain if parts of your experiment worked well. So the breaking open of the cell or other types in that case. So this actually is a good place for us to stop for today, because what we're going to talk about on Monday is Yes, today is Wednesday. Sorry. On Monday, we're going to actually talk about RT-PCR, which is going to allow us to look at this, but instead of using protein levels, we're going to look at the mRNA levels to see if we can get even more details about how the expression changes over time in those relevant. Have any other questions about Westerns, SDSPAGE, Affinity chromatography? I'm sure it buffered out when I asked if you had any questions. So Any other questions? Otherwise, you can email me or pop into office hours here in just a second. So I miss you guys tons. I will see you virtually here pretty soon. If you have a question, go ahead and pop it in the box. Otherwise, I will see you next week on Monday for RT-PCR.